Hi, my name is Rebecca Bruton, and I am an undergraduate student studying biochemistry. The topic I am presenting on is attention deficit hyperactivity disorder, commonly known as ADHD. I have a particular interest in this topic because I have a son with ADHD who also happens to have several related diagnoses, including a shared genetic monoamine neurotransmitter metabolic disorder. For simplicity, I will refer to monoamine neurotransmitters as MATs throughout this presentation. The purpose of this research project was to look at ADHD from a developmental biology perspective in order to gain a better understanding of this disorder. Through our literature review, we discovered that ADHD is predominantly a neurodevelopmental disorder that affects approximately 6 million children in the US. It is also predominantly a genetic disorder, with approximately 76% of cases associated with genetic abnormalities. ADHD does not typically present as a lone disorder. 70% of cases have at least one co-diagnosis, which commonly include autism, mood disorders, and learning disabilities. Children diagnosed with ADHD are also shown to have a significantly higher risk for developing Parkinson's disease later in life. Because ADHD is predominantly a genetic disorder, we decided to focus on the genetic factors that play a role in the development of ADHD and its related disorders. We found that many of these genes have connections to MATs. The first area of neurodevelopment that we explored was migration of neuronal cells. Two main genes associated with ADHD are included in the regulation of radial migration of neuronal cells. Radial migration is the migration of developing neurons from the apical region of the neural tube to the basal region. Disruptions in genes SLC6A4 and FOXP2 show increased levels of serotonin or dopamine and a decreased rate of radial migration. The next area that we explored was in axon formation and synaptogenesis. There were several ADHD genes associated with this area, including cadherin and protocadherin coding genes, CDH13 and PCDH7. These proteins act as short and long-range cues to guide axon growth. CDH13 knockout showed a decrease in dopamine and altered dopamine metabolic ratios, and PCDH7 proteins are signal transduction regulators of the dopamine D1 receptors. The other area we looked at was synaptogenesis. Loss of function in synaptogenesis regulating genes such as ADRGL3 show a decrease in dopaminergic neurons. During our research, we also found that there were rare disorders of the MAT metabolic pathway that were also associated with increased prevalences of ADHD. Disorders such as PKU and Sagawa's disease arise from a disruption in the body's ability to synthesize MATs. There are several genes associated with MAT metabolic disorders, one of which is the GTCH1 gene found in Sagawa's disease. The significant role MAT transporters and receptors play in the development of ADHD implies that the balance in synthesis and degradation must also be important. In the very early stages of neurodevelopment, most of the MATs are supplied maternally via the placenta, but by stage E10, the embryo must begin synthesizing them for itself. Given that disorders associated with a disruption in this balance also show higher rates of ADHD, perhaps a greater focus on this pathway and its disorders could potentially lead to a better understanding of ADHD across the lifespan. Current treatment focuses primarily on targeting MAT transporters and receptors, but perhaps more individualized treatments that include supplementation of MATs or their precursors could prove to be a better benefit with fewer of the psychological side effects.